Today, we are going to talk about enzymes. At any given moment, all of the work being done inside any cell is being done by enzymes. If you understand enzymes, you understand cells. So, let us tackle first the history of enzymes. There are two important people in connection with the discovery of enzymes. First is Anselme Payen, born on January 6, 1795, who was a French chemist known for discovering the first enzyme diastase and the carbohydrate cellulose. A diastase is any one of a group of enzymes which catalyzes the breakdown of starch into maltose. It was extracted from malt solution in 1833 by Payen. The name diastase comes from the Greek word diastasis, meaning a parting or a separation. It transforms starch into maltose and after that, it converts it into glucose. Next is Wilhelm Friedrich Kuhne, born on March 28, 1837, in Hamburg. He was a German physiologist. He is best known today for coining the word enzyme. Now that we have known the discovery of enzymes, we are going to talk about what is enzyme. It is a substance produced by a living organism that acts as a catalyst to bring about a specific biochemical reaction. So enzymes specifically are proteins that occur naturally in living organisms like plants, fungi, animals, and all living organisms. Enzymes are biological catalysts, which means it can speed up biochemical reactions in our body. So, without enzymes, these reactions take place at a rate far too slow for the pace of metabolism. In fact, the enzymes drive the biochemical processes that are necessary for life, for example, in case of digestion. It helps to break down large molecules from food that are present in carbohydrates, proteins, and fats into smaller molecules to make it easily absorbed like sugar, amino acid, and fatty acids. So, when the enzymes converted it all into molecules, it may instantly convert it into our necessary energies in our body. Enzymes are biological catalysts. What do we mean by catalyst? It is a substance that increases the rate of chemical reaction without itself undergoing any permanent chemical reaction. Therefore, it is an agent that provokes or speeds significant change or action, which enables chemical reaction to proceed at a usually faster rate or under different conditions than otherwise possible. But it only fastens the reaction and it will never have affected any chemical change. In the graph, you can see the word activation energy. What is activation energy? It is defined as the energy that must be added to cause molecules to react with one another. To start a chemical reaction, energy is required. It is the minimum amount of energy required to start a reaction. So in here, those who are under uncatalyzed reactions, which is the blue line, requires more of the activation energy so they can perform well what they intend to do. Unlike those who are under catalyzed reactions, in red line, they can perform well even with small amount of activation energy, as they already are an agent that can provoke significant change by themselves. What are enzymes made of? Enzymes are made from amino acids and they are proteins. And when an enzyme is formed, it is made by stringing together between 100 and 1000 amino acids in a very specific and unique order. There is a bacterium which possesses about 1000 different types of enzymes, and they are floating around in its cytoplasm at any given time. This bacterium is called Bacterium E. coli. And also, enzyme has its components. These are the simple enzyme and conjugate enzyme. First, let us know about simple enzyme. Simple enzyme, also known as apoenzyme, a protein component, is an inactive enzyme. Activation of the enzyme occurs upon binding of an organic or inorganic cofactor, a non-protein component. It consists entirely of proteins, which is polypeptide. Polypeptide is a linear organic polymer consisting of a large number of amino acid residues bonded together in a chain, forming part of or the whole of a protein molecule, and they contain no chemical groups other than amino acid residues. The next component is conjugate enzyme, also known as holoenzyme. Is composed of a protein portion called the apoenzyme and one or more non-protein portions. That non-protein portion is called cofactor. There are some metallic cofactors here like iron, copper, magnesium, calcium vitamins, and organic molecules which are coenzymes. So apoenzyme plus cofactor is equal to holoenzymes or conjugate enzymes.
Why are enzymes important? Enzymes are proteins that control the speed of a chemical reactions in your body. Without enzymes, these reactions would take place too slowly to keep you alive. Some enzymes, like the ones in your gut, break down large molecules into smaller ones. Others, like the enzymes that make the deoxyribonucleic acid or the DNA, use small molecules to build up a large complex ones. Enzymes also help cells to communicate with each other, keeping cell growth, life, and death under control. Now, how do enzymes work? Enzyme works to aid digestion and metabolism in the body. It is vital for our life and serves a wide range of important functions in the body. Some enzymes help break large molecules into smaller pieces that are more easily absorbed by the body, and these are protease, amylase, lipase, and cellulose for plants. Protease breaks down protein found in meats, nuts, eggs, and cheese. Amylase breaks down carbohydrates. Lipase breaks down fats found in dairy products. Other enzymes help bind two molecules together to produce a new molecule. Breaking molecules apart and putting molecules together is what enzymes do, and there is a specific enzyme for each chemical reaction needed to make the cell work properly. Enzymes are highly selective catalysts, meaning that enzyme only speeds up a specific reaction. Other types of enzyme can put atoms and molecules together. The molecules that an enzyme works with are called substrates. The substrates bind to a region on the enzyme called the active site. There are two theories explaining the enzyme-substrate interaction. In the lock and key model, the active site of an enzyme is precisely shaped to hold specific substrates. Unlike in the induced fit model, the active site and substrate don't fit perfectly together. Instead, they both alter their shape to connect.
The mechanism of enzymes. You can see the example of an enzyme which then have an active site and then a substrate, which is the sucrose, will bind to it to form an enzyme-substrate complex. And by this binding, it will then form products which are the glucose and fructose. The basic mechanism by which enzymes catalyze chemical reactions begins with the binding of the substrate or substrates to the active site on the enzyme. The active site is the specific region of the enzyme which combines with substrate. The central role of enzymes as biological catalysts. A fundamental task of protein is to act as an enzyme's catalyst that increase the rate of virtually all the chemical reactions within the cell. Although RNAs are capable of catalyzing some reaction, most biological reactions are catalyzed by proteins. Therefore, enzymes are proteins. Enzymes are biological catalysts that speed up the rate of biochemical of reaction in the cells, and chemical reactions that occur within the living organism are called metabolism. Now, The main role of enzyme is to act as catalyst and only speeds up the rate of reaction, and that's it. Let's now go to the factors that affect enzymes, which are the temperature and pH. Every enzyme works at its best when given a specific temperature. This temperature is called the optimum temperature of that enzyme. The optimum temperature of enzyme is 37 degrees Celsius. Reactions are accelerated when the temperature rises to a certain limit. But when this limit is crossed, naturation of enzyme takes place. Temperature and enzymes. The graph just shows that as the temperature increases, so does the rate of reaction. But very high temperatures denature enzymes. It shows that the typical change in an enzyme's activity with increasing temperature. The enzyme activity gradually increases with temperature up to around 37 degrees Celsius or body temperature. Then, as the temperature continues to rise, the rate of reaction falls rapidly as heat energy denatures the enzyme. pH. Changes in pH also alter an enzyme shape. Different enzymes work best at different pH values. The optimum pH for an enzyme depends on where it normally works. For example, intestinal enzymes have an optimum pH of about 7.5. Enzymes in the stomach have an optimum pH of about 2. So, these are all the things that you need to know about enzymes. Thank you for watching.